All right, our next presenter is Dr. Javier Bravo Cordero from the Icahn School of Medicine at Mount Sinai. Dr. Bravo Cordero is an associate professor in the Department of Medicine, Division of Hematology and Medical Oncology, and he's also at the Tisch Cancer Institute. Dr. Bravo Cordero was trained as a cell biologist and molecular biologist. He did his PhD in cancer biology at the Spanish National Cancer Institute, and then followed that with postdoc training in Dr. John Condilis' lab at Einstein. The work in Javier's lab really focuses on some very cool in vivo imaging technology and is focused on trying to understand how tumor cells disseminate during metastasis. So the title of his talk today is Extracellular Matrix and Dormancy. Welcome, Javier.
Thanks so much, Elana, for the introduction, and I really want to thank the organizers for giving me the opportunity to present here the work on my lab. I'm going to tell you a little bit about the work that we've been doing trying to understand how the extracellular metrics regulate this process of dormancy we just heard about, and how we can use that biology in order to really control the fate of these disseminated tumor cells. Oops, let's see how this works. So in breast cancer, metastasis is the primary cause of death in these patients. And what we know is that metastasis is a very complex process in where the tumor cells have to undergo multiple transitions. They have to invade into the stroma, intravasate and extravasate from the blood vessels, and eventually see that at distant sites. What we know is that these disseminated tumor cells don't restore proliferation immediately. They enter in this period of quiescence that can last for many years before these tumor cells awaken and start proliferating. So my lab is really interested in understanding this biology and how tumor cells sustain this quiescent state in distant sites and how they transition to awakening. So in the context of breast cancer, what we know is that very early during tumor progression, there is early disseminated tumor cells that can leave these early lesions and see distant sites. And when patients are treated and the primary tumor is removed, these patients that are in minimal residual disease, we know that unfortunately 5 or 10 years or 20 years later, they develop recurrences. And because of this early dissemination effect, what we know is that these cells that see that distant site enter in this period of quiescence, and they are the consequence of this relapse that you see in this purple line. So a lot of biology is being built around these dormant cells, trying to understand how they sustain this phenotype. And the work from many labs have defined these dormancy hallmarks in order to identify and characterize these cells. So we know these cells are deeply quiescent, they are in G0 growth arrest, they activate survival pathways, and they have latent pluripotency. So interactions with the extracellular medicine, with the tumor microenvironment, can trigger the awakening of these cells and sustain that phenotype. So what my lab is trying to do is follow the fate of these cells during tumor dissemination, and we utilize different types of imaging technology that allow us to visualize tumors and metastatic sites in real time and really get temporal information on how these cells behave. So we use a technology that is called lattice light sheet microscopy that basically allows us to clear the tissues and perform volumetric imaging, as you can see in this example in the mammary gland, where we can clearly see where the epithelial cells are displaced. In this case, they express a GFP, and we can have a closer look at these early lesions that are formed in these mammary glands. And in order to understand the behavior of these tumor cells, what we apply is intraviral tooth photon microscopy in order to really understand the behavior and how these tumor cells interact with the extracellular matrix. So you see the movie on the right, you see a breast tumor and these early disseminated tumor cells invading into the stroma, and in blue you see the collagen matrix, which is the ACM. So we've been very interested in understanding how the extracellular matrix actually control this, the fate of these disseminated tumor cells. So as you know, the extracellular matrix regulates many stages during tumor progression, from tumor development, invasion in where the matrix has to be remodeled, and metastatic outgrowth. So the composition of the extracellular matrix in tumor and metastasis come from the stroma, from the fibroblasts, but also the tumor cells are very active producing matrix. And the molecules that are within these matrices contribute to the stiffness and the three-dimensional organization of these matrices. And all these are going to control control the fate of these tumor cells. And also very important, which receptors these cells express in the membrane is going to determine their fate and how they interpret the different matrices as they disseminate from the primary tumor to the distant sites. So the question that we've been trying to understand is whether these disseminated tumor cells can control their fate by regulating the composition of the extracellular matrix in the three-dimensional architecture. So the work that I'm going to show you was done by Julie Di Martino, a very talented postdoc in my lab. She wanted to really understand the dynamic of the extracellular matrix during tumor dissemination and tumor dormancy and awakening. And what we start doing is we generate a series of cell lines. We work in head and neck models and also in breast tumor models. And we engineer these cells to express a G0 state biosensor that basically allow us to identify that are the cells that are quiescent in vivo. So this biosensor accumulates in the nucleus, and the accumulation identify the cells that are in G0. So what you see here is intraviral imaging of a primary tumor, in where we can see that this, the distribution of the sensor is cytosolic in the cells are cycling. So when CDK2 activity is high during a cell cycle, the sensor can release from the nucleus and remain cytosolic. So you can see we can identify cells in mitosis, and we also have like a H2B fluorescent molecule that allow us to identify the nucleus for thresholding. So what we did is we removed these tumors, and then we implanted a window in order to follow these cells over time. And we know that after tumor recession, there are some cells that are left after surgery, and also there's local invasion of cells within the stroma. And these cells enter in quiescence after surgery, and they last for up to 5 to 10 weeks. So when we perform intraviral imaging of these cells and look at the spatial distribution of the sensor, what we found is that all the cells localize the sensor in the nucleus, meaning that these cells are in deep quiescence. But what was very interesting was when we start analyzing the extracellular matrix around the cells that you see here in gray. So we start seeing this highly disorganized matrix and wrapping up the cells in vivo. And when we follow the cells over time and these cells spontaneously awake, we see the shift in the architecture of the extracellular matrix from this very disorganized Carli matrix that are more aligned. So we think that these transitions of extracellular matrix dynamics actually are playing an important role in regulating this dormancy to awakening transition. So we really wanted to understand how the tumor cells, these disseminated tumor cells, contribute to this particular extracellular matrix. So in order to do that, we've been using different tumor models in head and neck and in breast that represent proliferative and dormant states, like the ones you see here. So these dormant cells, when implanted orthotopically, they don't form tumor, or they remain as a single cells in the lung. And you can do quantitative imaging analysis of the extracellular matrix around the cells, and what we found is this shift in the alignment of the matrix with a decrease in fiber orientation in these dormant tumor cells. And similar dynamics we observed in mammary tumors in 41 model and the D2, which are breast tumor models that also have the dormancy counterpart. Now, this is how this matrices looks like. They're very disorganized. They have a very particular three-dimensional organization. So we decided to go a little bit more deeper and really understand what is the composition of this matrix in order to identify the drivers of dormancy in the ECM. So we collaborated with Alexandra Navas. She's assistant professor in the USC, and she developed this matrisome platform in order to profile the extracellular matrix and tumors and metastasis. So this is the distribution of all the matrisome associated and matrisome genes within the human genome. We have the glycoproteins, collagens, proteoglycans being part of the core matrisome. And what we did with her is perform enriched proteomics for the extracellular matrix and do quantitative analysis in order to define this quiescence of these dormant cells. What are the extracellular matrix that are produced by these dormant cells? And what it came out was very interesting. What we found is that 55% of the matrix produced by these dormant cells are collagens. They're highly enriched in collagens, and we see a shift in the type of matrices that they produce as they start proliferating, with a decrease in collagens and increase in proteoglycans. So we validate this in head and neck models and breast cancer models. As you see here, this enrichment of extracellular matrix around these dormant cells in tissues. Now, what we were looking for are molecules within the extracellular matrix that are driving this quiescence. So what we look at the collagens that the tumor cells produce, what we found is that collagen 3 was the most abundant extracellular protein secreted by these dormant cells. And collagen 3 is a type of fibrillar collagen that form triple helix and organizing fibrils in the large fibers with collagen 1, as you see in the movies I've been seeing you. So we see this increase in collagen 3 in breast tumors, in the cells that are dormant, and in the tissues you can see this nice enrichment of collagen 3 fibers in around these dormant cells that is completely avoid depleted in these proliferative counterparts. And if we look for disseminated tumor cells, we can see that similar as I was showing you at the beginning, we see this collagen 3 enriched around these dormant cells, and again, when the tumor cells awake, there is a depletion in collagen 3. So there is a very interesting dynamic of the content of collagen 3 during dormancy and awakening. So we knock it down, and what we found is that these cells spontaneously start proliferating. We get this increase in tumor proliferation, increase in proliferative markers as phosphohistone 3, and decrease in very important dormancy markers like NR2F1. So this, we also observe this phenomenon in breast tumor models, in dormant cells in breast, in where depletion of collagen 3-1 drive the awakening. And we did these experiments in CAM assays, but we also did some mouse experiments doing tailbone injection, seeding the cells in the lung, and similarly, when the cells lack collagen 3, they are not capable of sustaining this quiescence anymore and start proliferating and forming large metastases. Now, what was very interesting is how is this particular collagen regulating the fate of these cells. So we did a series of imaging experiments in taking cells and putting them in different type of matrices and look at the status of this biosensor that I was showing you at the beginning. So if we take dormant cells and we put it in collagen 3, we see this accumulation of the sensor in the nucleus, it's in G0. Now if we take a proliferative cell and put it in collagen 1, the cell is actively migrating, they degrade the matrix, they divide nicely, but if we move them now and put it in collagen 3, what we start seeing is that these cells now start wrapping themselves up with a collagen 3 and start accumulating the sensor in the nucleus. So we're pushing the cells into this quiescent state. So with this idea in mind, we ask ourselves, okay, can we reprogram the ECM microenvironment and push proliferative tumor cells into this quiescent state? So we did a series of in vivo experiments in where we inoculate tumor cells with or without collagen 3, and then we monitor tumor growth. And what we found is that in the conditioning where we have enriched for collagen 3, we see this interruption of growth, and the tumors grow slower. But what was very interesting also that we found is that if we do a similar experiment as I showed you at the beginning, we remove the primary tumor, and at the time of resection, we treat the wound with this collagen 3 scaffold that are sponges filled with collagen 3, and then we monitor for relapse. What we found is that the animals that were treated with these collagen 3 sponges actually have a lower recurrence, so suggesting that we're pushing the cells left behind into this quiescent state. And we kind of validate that by doing intraviral imaging at the end of these experiments and looking at the state of the sensor. The remaining cells in that areas are in quiescence. Now, how are these cells, the dormant cells, interpreting these matrices? So there is different type of collagen receptors, integrins, DDRs. What we found very interesting is on all of the dormant models that we were studying, we see this upregulation of the DDR1 collagen receptor in the dormant cells when we compare with the proliferative ones. So this is a very exciting an interesting molecule. We also found that these dormant cells are very addictive to collagen 3, and this addiction is dependent on DDR1. So we can knock down DDR1 and prevent the binding of dormant cells to the collagen 3. And so we thought that DDR1 would be playing a major role in controlling dormancy of these cells. We did, we knock it down and then put the cells in the lung, and what we found is that the cells, again, they're not capable of sustaining dormancy anymore. They start proliferating and forming metastases. How is DDR1 regulating the dormant state? Well, it's doing it through regulation of the matrixome. So we found that as this tumor awake in the absence of DDR1, we see this shift in the architecture of the extracellular matrix. And when we did gene expression analysis, what we found is that the matrixome genes start becoming more similar to the proliferative tumors that we were analyzing. And the proteomics actually show us that indeed there is a drop of collagen 3 in these awakened tumors, and we see a new remake of the entire matrixome with new collagens being deposited in these metastases, like collagen 22 that wasn't present in the dormant tumor. So we think this dynamic of the extracellular matrix during dormancy and awakening is actually pointing to a specific targets within the extracellular matrix that may be controlling this awakening phase. Now, the last piece I want to share with you is how DDR1 is controlling the matrixome. We found that in the absence of DDR1, there is then regulation of different transcription factors. We found that STAT1 is actually highly downregulated in the absence of DDR1. And if you knock down STAT1, the mRNA levels of collagen 3 drop. So STAT1 is involved in regulating expression of collagen 3A1 and also regulates dormancy of the cells. We can knock it down again, and tumors start, the tumor cells start proliferating again. And there is some changes also in the expression of the DDR1 receptor. So we think all these three components are part of a signaling loop that feed the cell in this ECM-driven dormancy. STAT1 gets activated in the presence of collagen 3. We did some experiments looking at the translocation of phospho-STAT1 in the nucleus in the presence of different extracellular matrices. We see this translocation in the presence of collagen 3, and similar to DDR1 and collagen 3, we see this awakening of the tumor cells when we deplete STAT1. So just to finalize, what I want to give you as a take-home message is that we think that these dormant cells are capable of constructing these cocoons enriched in collagen 3, and they're very dynamic and actively deposit these tumor cells in order to these matrices in order to sustain their quiescence. And if we interrupt this dynamic between the dormant cell and the extracellular matrix, we can trigger the awakening of these cells by either downregulating DDR1 or affecting the collagen 3 levels in vivo. So we think that the collagen 3 is a very important marker and regulator of dormancy. I'm sure that many other collagen molecules will be shown to play a role, but it was very interesting to see that this collagen was already described in the 90s as a gene that gets regulated with fibroblast centers in quiescence. And more recently, work by different labs have shown that in breast tumors, in HER2 and ER-positive tumors, the disseminated tumor cells that leave early these lesions in these mouse models actually overexpress collagen 3. So we think that the biology that we uncover is playing this, the functionality of this collagen in these early disseminated tumor cells. So with this, I will just finish and thanks my lab and the people that did the work. As I showed you, this was done by Julie Di Martino, and also we have an excellent group of collaborators, Julio Guirreguizo and Alexandra and Elana, and also the funding agencies that generously support our work, and also the Advanced Bioimaging Core at Sinai, where we did all this work. And we're actively recruiting, so if anyone is interested in pursuing some of these projects and work on dormancy and in the context of breast cancer, performing imaging, so feel free to email me, and I'm happy to take any questions.
That was a fantastic talk, Javier. It looks like we have one question here.
Yeah.
Compliment for your talks. Just coming back to the role of extracellular matrix on dormancy, what is the role that you assign to the cancer-associated fibroblasts secreting a lot of protein belonging to extracellular matrix on the dormancy?
Yes. No, that's a very interesting question. So the fibroblasts play a major role in depositing in matrices. So when we analyze the matrixome at the protein levels. We also look at how the proteome of the fibroblast is affected by dormant cells and proliferative cells, and we see a communication between them, so that the dormant cells are capable to reprogram the fibroblast, for example, to also start secreting more collagen 3. So we think that within the tumor microenvironment around these cells, the stroma may be instructed also to secrete some ECM molecules that sustain the dormancy of these.
You mentioned also STAT1 secretion, but cancer-associated fibroblasts do secrete also STAT3.
Yeah.
So maybe that both may converge.
Yeah, that's very interesting.
Thank you.
Thank you.
Ian Kunkel, University of Edinburgh. In head and neck cancer, the primary tumour often remains in situ and treated by radical radiotherapy. Do you see any evidence of these quiescent cells in the primary tumour, or is it an acquired state which only is relevant to metastasis?
We do see some quiescent cells within the tumor, but the population that we are analyzing is after tumor recession. So that's in the models that we have when we remove the primary tumor, the cells that are left behind or that already have undergo local invasion, they enter in quiescent for a period of 5 to 10 weeks. So we think it's in that moment in where they utilize these mechanisms to remain dormant there.
Thank you.
I think in the interest of time, we have to move on because we have another session in here right after.